Hello friends, this is Jim with Science Talk. Wanted to share with you an interesting story. And the title is How 250 Siberians Became the First Native Americans. Americas, of course, North and South are fairly large continents. But the Native American group that first settled it was very small. Estimate is only about 250 people. This is based on a new genetic study. These people are known as the founding group because they founded the first population, migrated from Siberia to the Americas about 15,000 years ago. Of course, at that time, sea levels were lower. There was ice age that was happening, and you had the Beringia, so basically, it was a big, huge landmass from basically the northeastern portion of Russia over into Alaska, and they traveled that route there. So, one of the lead researchers, Nelson Fagundes, is a professor in the Department of Genetics at Federal University of Rio Grande do Sul in Brazil. Figuring out the size of the founding groups is key because it determines the amount of genetic diversity that gets passed on to the group's descendants. Notice the genetic potential. That in turn could alter how effectively natural selection weeds out bad genes. The more diversity, the more likely bad genes get removed, or to use the parlance, selected out, selected against. Large populations have very efficient selection, while in small populations, mildly deleterious alleles. Allele is a form of a gene. To use simple Mendelian example, okay, if we have a gene that determines the height of the plant, we have a tall gene, or the other version of that gene for height would be short. So we have a gene that determines the height of the plant. One allele makes it tall, another allele makes it short. And tall might be the dominant allele. So as long as there's one dominant allele, that trait gets expressed. The recessive allele gets masked. Unless we're talking incomplete dominance, but that's another topic. So again, small populations have mildly deleterious alleles can spread. So if you have a large population, any deleterious alleles get selected against and they're removed. Smaller populations, they persist. Why is this bad? Well, it increases your susceptibility to diseases. To investigate the size of the original Native American founding group, Fagundes and his colleagues studied DNA samples from 10 Native American individuals scattered across Central and South America. 10 people from different Siberian groups and 15 people from China. The Native American groups included the Ache of Paraguay, the Bribri, the Guatuso, the Guami of Costa Rica, the Lengua of Argentina, the Quichua of Peru, and the Arara, Waiwai, Sabante, and Zoro of Brazil. Now you're going to say, what about native folks from North America? Why are they not represented? Well, they did not include Native Americans from North America for the simple reason that many of them formed unions with people from later migrations. Thus, this makes it more difficult, a little mosquito here, makes it more difficult to exactly pinpoint the group. In fact, it makes it more difficult to pinpoint the genetics because there's been so much mixing added variation in diversity makes it harder to separate out the genetic markers. So what they did was they looked at 9 regions that contained about 10,000 base pairs, the ATGC base pairing. Researchers know that genetic variation within a sample is directly related to population size. Increase the population size, increase the genetic variation. Directly related. Decrease population size, decrease the variation. So that fact combined with also that genetic divergence between 2 populations, Native Americans and Siberians, as in the case with this study, will increase with time. In other words, the further 2 populations are separated by time, the more time that lapses between their separation, you're going to see increased genetic divergence. And you can actually sort of like use that as some sort of a timing mechanism, or at least a predictive mechanism. So they took this information, the DNA data, they shoved it into the computers, ran the simulation models, and they worked backward to figure out the original size of the founding group. The models found between 229 and 300 people. And then they finally, for some reason, settled on about 250 people. Now that's a small number. And that could lead to what's called a genetic bottleneck. Meaning, what's a genetic bottleneck? Little genetic variation associated with the first major migration wave into the Americas. Bottleneck. You have very little genetic variation. And when you have something like that, the chances to acquire genes to help survive in a new situation are reduced. In other words, this can lead to the increased probability of death due to not being adapted enough to your environment. Now, as Dr. Fagundes says, the 250 number is a guess. It's an estimate. However, this estimate is similar to findings of other studies, so there is support for this value. Estimating the size of the genetic bottleneck is important because it helps scientists figure out how many genetic markers are needed to capture the genetic diversity of North American populations in studies, as well as evaluating the harmful or beneficial different versions of genes that are present. So you can construct important genetic information, genetic profiles. And you might be able to construct the genes that, propensity to code for, say, certain diseases, and then take steps to deal with those. The genetic data illustrates how ancient migration unfolded in the Americas. This is a statement from the other co-researcher, Michael Crawford, who is a professor of anthropology at Kansas University. Native Americans would settle in a new place, and as the population and, in essence, fertility grew, people from one population would break off, form another population in a neighboring area. And you just continue this process. So then after 15,000 years, next thing you know, you're in Argentina. The study was published in the May 1st issue of the journal Genetics and Molecular Biology. So, this is very fascinating. And when you think about it, if they turn out to be correct, we had this small group of 250 people, and it gave rise to all the native groups we see across both continents, North and South America. That, yes, over 15,000 years is what do we expect to see in the genetic diversity? Well, 15,000 years in geological terms is not very long. If we had more than 250 individuals, chances are very, very high, that the genetic diversity would be much more varied. But with such a small group, again, we're using the bottleneck concept, the genetic diversity is not that great. So we can, and this is what they did in the study, they looked at the gene markers, and then they can tell how much separation, how different they are from each other, separation over the 15,000 years. It's just an interesting study. I find studies dealing with Native Americans in the Americas, you know, the migration paths, how the societies was, and so on and so forth. I find it to be very interesting. Here in Alaska, we have the Athabascan, we have the Yupik, the Inupiats. We don't call them Eskimos here. And we have other, then we have the Tlingits down in the southeast. So they're the Aleuts, where we get the Aleutian name for Aleutian, the Aleuts out in that direction. So we have a lot of native groups here in Alaska, and this kind of helps us better understand how, you know, they came here and how they came about, how they moved about, and just a general understanding of their biology. So anyway, I hope you found this interesting, and we'll talk soon. Thank you for your time. Hey folks, just a reminder to please subscribe and click the bell so you know when I drop a video. And please share my work with others. Please tell them of my channel. And I hope you'll become a patron at Patreon. I place videos on there from time to time exclusively for my patrons there. If you're already a patron, thank you for your support and continued support. Details in the box below. Thanks.